focus is particularly on the phase behavior of ritonavir in copovidone and aiming to understand how it dissolves or releases while maintaining supersaturation, which is crucial in its therapeutic effect. And to achieve this understanding, we employ advanced molecular modeling techniques, including molecular dynamics and dissipative particle dynamics. And this approach allows us to simulate the complex interaction between the dispersion at a molecular level. By leveraging these thermodynamic principles, we were able to describe and to predict the behavior of ASDs, which is critical addressing the challenges associated to these purely soluble drugs. To understand this release mechanism, or when it comes to loss of release, we identified two loss of release main driving forces. The one is when we have a fast crystallizer. We call this loss of release mechanism 1. There, what can happen is that at a certain high drug load, crystallization-induced loss of release happens. This means API is supersaturated and crystallizes very rapidly directly at the interface between ASD and water. And this affects the release negatively because when this happens, the API cannot erode in the amorphous state anymore into the media. And the loss of release mechanism 2 is the one when we have, for instance, a slow crystallizer, but the one which shows large miscibility gap, so tends to liquid-liquid phase separate strongly. There, in this mechanism, phase separation is the trigger. So beyond a certain drug load, the mixture undergoes phase separation at the interface, and the API-rich phase, which is formed, is often additionally below TG, so shows a very low mobility and stays with the tablet. So also there, we have a very poor release performance then. And to highlight here is really that the thermodynamics help a lot understanding the driving forces for the system. So with that, you really see what can happen. But still on the molecular level, the drug load-dependent release mechanism on the ASD is not fully understood yet. And this is why we reached out to Schrödinger, and with Schrödinger, we had in molecular simulation very interesting findings on that.
The drug that we're studying is ritonavir. This is a drug that is used for treatment of HIV and is a drug that is already marketed as amorphous solid dispersion. Has low aqueous solubility, high lipophilicity, and a tendency for polymorphic transition, so the perfect candidate for amorphous solid dispersion. And then the second ingredient of our formulation is copovidone. This is a well-known polymer for amorphous solid dispersion. What I will be talking about is what we call molecular modeling, so molecular simulations, which are trying to connect or to understand the behavior at the macroscopic level by looking at the system at the microscopic level, so looking at how the different molecules interact. So from a general overview, you can already see that only changing the drug load, we have different behavior in the dissolution mechanisms. And for example, if we look at the lowest possible drug load, 1%, by the end of the simulation, we have a quick and total disaggregation dissolution of the system in water. So both the polymer chain are released, but also the drug molecules are released in the solution, and they are released as individual molecules. If we move to higher drug loadings, so between 5 and 20% weight, for example, we still have disaggregation of the amorphous solid dispersion for both the polymer and the drug. But the drug, you can see that start forming some sort of clustering between themselves. So you have these classes of drugs that are formed during the simulation that are obviously increasing with the increase of drug loading. And finally, if we switch to the last system where we have 40% drug load, you can see that the overall dissolution kinetic is slowed down, so neither the polymer or the drug fully disaggregates. Details, you can see that at the beginning of the simulation, there's a clear interface between the dry amorphous solid dispersion and the water, and by the end of the simulation, the amorphous solid dispersion the water has entered the amorphous solid dispersion and the dissolution has started. And in particular, you can see different layers within the models. We can also define some metrics and follow them for different formulation at different points in time. So what I'm showing here is the number of remaining molecules, which means all those drug molecules that do not dissolve enough to reach this sink region where they would be considered dissolved and converted into water particles, and how this number is changing with at different points in time for the simulation. Here again, we can define some more quantitative metrics. So here we're looking at the concentration, the number of drug molecules inside the main cluster at equilibrium for different drug loads. And you can see that it increases linearly with the drug load, which means that the more drug molecules you put, the bigger the cluster forms. And it's just one cluster rather than different clusters, for example. And here is the radius of gyration of the cluster as a function of drug content. This is a measure that helps us understand the size of the cluster, but also how compact it is. And you can see that it goes down with increasing the drug content, and it sort of plateaus after the 20% weight, which means that from this value on, the cluster is really well defined and compact, and is mainly controlled by the drug molecules. One other thing that we can get easily from simulations are the interactions between different molecules or portions of the system. So I'm not showing all the results here, but we could look, for example, which part of the drug is interacting with which part of the polymer and how different parts differ. One thing that I'm highlighting here is are the contacts, so the interactions between the drug molecules and the water particles, normalized by the number of drugs in the system. And with increasing drug content, these numbers goes down, which means that the more drug molecules we put in the system, the less these drugs molecules are in contact with water. And this is mainly due to the fact that this cluster, this phase separation forms, and therefore all those molecules that end up in the middle of the cluster are really not in contact with water anymore. Some of these insights can be connected to results from thermodynamic modeling and experiments. And so I'll hand it over to Stephanie again to show what they found on the AbbVie side.
Yes, thank you, Irene. Okay, super. So on the left-hand side, you see the predicted ternary phase diagram, and I will keep it very short now, but the key message is it aligns very well with what we see from the MD, because at the bottom of the ternary phase diagram, you can see from left to right the increasing drug load and how different the hydration pathway through the phase diagram is. And we see that, yes, for the 40% drug load, we enter the LLPS at the very early stage. We see from these high lines, the driving force for LLPS is very strong. The amount of API enriched in this LLPS phase is increasing with drug load and very strong for 40%. In this study, we examined ritonavir with co-povidone, amorphous solid dispersions, and our research was a combination of thermodynamic modeling and molecular modeling to unravel the nanoscale processes that dictate really our ASD release performance. And our critical findings were that at higher drug loads, specifically between 20 and 40%, the system tends to strongly undergo phase separation, leading to these drug-rich clusters. And our predictive models were tested and confirmed experimentally with microscopic erosion time tests. And the integration or the combination of thermodynamic modeling, these MET images and MD really gave us some profound insights in the interaction, what really comes into state when the ASD releases into the aqueous phase. And by leveraging these modeling and simulation, we can enhance our ability to predict drug load dependent dissolution behavior, which is a critical step towards like finding the right drug load for an ASD.